Okay, it looks like we have had everyone log in here. So I will go ahead and get us going. Again, thank you so much for being here this afternoon with us for our monthly Know Your Ocean speaker series. My name is Jill Wirt, and I will be your MC for today. I am a project manager with Maui Nui Marine Resource Council, and today we are joined by Dr. Simon Freeman, who will be presenting about signals in noise, adventures in making use of natural sounds underwater. Maui Nui Marine Resource Council is celebrating 15 years of working for clean ocean water, healthy coral reefs, and abundant native fish. Our work wouldn't be possible without the support of viewers like you. Today's presentation is part of Maui Nui Marine Resource Council's monthly Know Your Ocean speaker series, usually held on the first Wednesday of each month at 5:30 p.m. via Zoom. Obviously, today's an exception. This monthly speaker series is supported by the County of Maui. Before we get started, I wanted to share a few announcements from Maui Nui Marine Resource Council. Breaches and Brews Science Nights are back tonight. Please join us at Kohola Brewery at 5:30 p.m. to hear from our very own Chief Scientist, John Starmer, talk about what watersheds are, why they matter to marine conservation, and why in the world a marine-focused organization like Maui Nui Marine Resource Council spends time supporting efforts to build fire breaks, plant vetiver grass, and put up deer fencing. We will see you all there. Maui Nui Marine Resource Council recently completed their initial sampling of intertidal limu, or marine algae, to evaluate the influence of pollution on coastal ecosystems. Samples were sent to Hilo's Analytical Laboratory for nitrogen isotope analysis. The abundance of specific nitrogen isotopes provides clues to whether the fresh water flowing into the nearshore environment is relatively clean or under the influence of pollutants that enrich nitrogen, such as sewage and inorganic fertilizers. Stay tuned to see the results. Is coral restoration coming to South Maui? Over the past few weeks, MNMRC staff and partners from Oahu scouted potential locations and captured photo mosaics of potential areas to begin coral restoration in South Maui. Thank you to Maui Pacific Divers for lending their boat and crew so that we could get out on the water. If you are a South Maui resident and want to be involved in the selection process for potential sites, you can email info@mauireefs.org. Hui o ka Viola is celebrating its 100th South Maui sampling event this month on July 17th. Congratulations to the Hui o ka Viola team for their hard work and dedication to collect quality assured data that is used by the Department of Health Clean Water Branch. A few notes before we introduce our speaker. We will leave time at the end of the presentation to answer your questions, and we invite you to submit your questions by using the Q&A button on the lower edge of your Zoom screen. This presentation is also being recorded and streamed to Facebook Live and will be available on our YouTube and Facebook profiles. If you're viewing live from Facebook, you can submit your questions as a comment, and then they will be relayed to me at the end of the presentation. And now for our speaker. Dr. Simon Freeman serves as a program director at the Advanced Research Projects Agency under the Energy sector, ARPA E. His focus at ARPA E is on maritime energy and environmental sensing technologies. Freeman previously worked as a civilian oceanographer for the U.S. Naval Research Laboratory and the Naval Undersea Warfare Center Division, Newport. Freeman was a principal investigator in the development of inflatable and bioinspired soft robotic systems for underwater vehicle and sensor design, new signal processing methods for acoustic sensing, detection and environmental exploitation, and pressure-tolerant technologies to enhance our access to the seabed. Freeman earned a BE with honors in mechanical engineering and a BS in marine science from the University of Auckland, New Zealand, and a PhD in oceanography from Scripps Institution of Oceanography. Simon, thank you so much for being here. I will let you take it from here.
Thanks so much, Jill. And thanks for the invitation to be here. It's a pleasure to address you all. If you'll bear with me for a moment, I'm just going to share my screen. see. There we go. Can you see that presentation okay there?
Yes.
Awesome. All right. Well, I'll go through this, and there are a couple of videos. I might have to exit out, show you the videos, not in presentation mode. But look, thanks for the opportunity to speak to you all this week. I'm going to talk about signals and noise, our adventures in making use of natural sounds underwater. And this is work that my wife and I, so Lauren and myself, worked on in graduate school, and then afterwards working for the Navy. I want to acknowledge some of the folks who have helped make this happen. My wife and I both went to graduate school at Scripps Institution of Oceanography, part of UC San Diego. When we worked in Hawaii, we worked with Mark Lammers at the Oceanwide Science Institute. This is back when they were based in Kaimuki in Oahu. And I recall Mark shortly afterwards moving to Maui. NOAA and the Rapid Ecological Monitoring Team at NOAA well, back then it was Koala Basin, now it's Ford Island in Oahu, helped us out. We went on one of their research cruises. And of course, we need to acknowledge ONR, the Office of Naval Research, who funded a lot of our work. So firstly, before I begin, I want to talk to you about what a soundscape is. This is something which we experience every day when we go outside and listen to the environment. And it's like a landscape, but you're listening to it, a soundscape. And just by listening, you can understand a lot about what's happening around you. You know, whether there are cars driving up and down the street, whether it's windy, whether it's raining, whether there are people around, etc. The same thing works underwater, you know, but under the sea, it's a very different place acoustically, where sound propagates in a very different way. There's a wonderful analogy I saw at some point where, you know, someone said to me, if you stand on top of a mountain and you look down upon a valley below, you can see for miles, but you can hear maybe 20 meters. Some bugs maybe flying around you, etc. But you can't really hear beyond that. Under the ocean, the opposite is true. If you stand on an undersea mountain, you can only see about 20 meters tops, but you can hear for miles because sound is the most efficient radiation underwater, far more efficient than light or anything else. So one of the challenges that we sought to address was this problem of how to survey remote marine areas. So Hawaii is a state that is blessed with a lot of these outlying islands. You have the Papahanaumokuakea Marine National Monument. You have tremendous natural resources there, but understanding what's happening in these remote places, any changes, it's really difficult to do. And today it's done, this monitoring is done through a laborious process where ships go out, divers get in the water, scientific measurements are made, etc. It's very expensive, and you can't always see all of the organisms underwater when you scuba dive. Sometimes they escape, sometimes they hide. You could use satellites, but remote sensing is subject to being obscured by clouds. You can't do it at nighttime. There are some issues with resolution and atmospheric correction. So there's this question, you know, can we listen to the soundscape in these areas? Passive acoustic monitoring. It's a way that we could quantitatively, let's say, count the number of fish grunts, right? An acoustic recorder integrates what happens within acoustic range. It's a spatial integrator. And remember, sound travels much more efficiently underwater. So a single recorder can survey a much larger area than the equivalent situation on land. And acoustic recorders are pretty inexpensive, so much less expensive than a ship, for example. So could we do this? But there are some caveats. Biological sound sources are largely unvalidated. We can hear all sorts of noise, but what's creating it? We don't really know. The ocean is a very echoey place. There's a lot of reverberation and what we call channel effects. So how sound is modified between a source and a receiver. Like, does it bounce off stuff? Does it partially get absorbed by interaction with the seafloor, etc.? That's the channel, right? And then large variability in space and time. So if you snorkel over a coral reef, you see patches of sand, patches of coral, some living things, some things not alive. So you've got a lot of variability in sound sources. So the goal here is if we could extracting ecologically meaningful information from soundscape recordings. And so this process should be fairly straightforward. You deploy some hydrophones or underwater microphones, collect information, some stuff happens, and then you get inexpensive real-time ecological monitoring. And this is something that we do on land already. There's a map here, global ecological zones. This is satellite derived. We do this on a large scale on land already. It's just that we can't use satellite data sort of deep under the ocean beyond about 10 or 15 meters. So the question mark box is the difficult part, right? The details really matter here. And this talk is about creating experiments and trying to solve some of these mysteries specific to this how section. There are lots and lots of things that we could do here, but I'm only talking in this talk about the biological sources in the immediate area of the hydrophone from little critters that you find on the reef. Most of these are poorly characterized. I'm not talking about human-made noise right now. Neither am I talking about marine mammals. And then noise from non-biological sources, so breaking waves, wind, earthquakes, etc. That's sort of outside of scope. And then we're not really discussing during this talk the impacts of scattering and reverberation. But I'll get to how that's mitigated in a moment. So you may ask, what's it like down there? I'm sure many of you have been snorkeling, but if you haven't, it's a very complex world. Doesn't necessarily look like an aquarium. There's a photograph here of some of our scientific instruments deployed on a reef, and you can see the sea floor is covered in coral. And there aren't huge numbers of fish that you may immediately expect. So the variability of where the fish are present, it changes with time and place, right? But it's a very large area, and I can't really depict to you what it may sound like. I don't have any audio recordings with me today, but for the most part, when we look at acoustic data, we look at it visually. Like, humans are really good at looking at understanding the difference between colors and picking out details in an image. And so a lot of work in acoustic processing is done through looking at the pictorial representation of sound. And that's done through what we call a spectrogram. And so on the left here is a representation of coral reef sound. I'll talk a little bit about how a spectrogram is arranged. I'll show a lot of these during this presentation. So I want you to know how to interpret them. So firstly, you see a lot of color, and that color represents acoustic intensity, or the amount of energy in sound, where red is more and blue is not much. This is a logarithmic scale, decibels, and decibels have been developed for characterizing sound because the amount of energy in sound covers such a wide range. Our ears are very sensitive to the smallest amounts of acoustic energy to lots and lots of acoustic energy, right? The scale is enormous. So that's a logarithmic scale where, let's say, 63 dB is twice the level of 60 dB. Right? So it's a slightly not intuitive to understand, but by and large, you could think, well, red is more energy, blue is less. On the horizontal axis of this picture spectrogram is time. This covers a 24-hour period of ambient noise on a Hawaiian coral reef. And you can see there's two dashed vertical lines there. I'll get to those in a moment. The vertical axis represents frequency, and that spans from 0 to 20 kilohertz. That's about the range of human hearing. So a child can hear perhaps up to 20 kilohertz, and we can hear down to about 20 hertz. And that's such a small number that it doesn't really show up very well on this plot. But so now I've explained the axes, I can step through what's actually in this spectrogram. So you can see that the color, if you look up the vertical axis, it's very cool below about 2.5 kilohertz. And about 2.5 to about 15, you have this sort of yellow reddish blob during the day. And then above 15, it's kind of green, so there's less energy. At nighttime, things really change. So those dashed lines represent sunset and sunrise. When the sun goes down, the sound goes up. So things get much redder, especially between sort of 3 and 7 kilohertz. That's the sort of darkest red area. This represents a nightly chorusing of animals on the coral reef. And that chorusing continues until sunrise. And like clockwork, it becomes quiet when the sun comes up. This is a typical spectrogram, a typical description of what happens acoustically on a coral reef at night. So it's strongly diurnal. And then you have seasonal and lunar cycles that influence the soundscape as well, but typically at low frequencies, so less than 1 kilohertz, which is kind of, you can't really see it in this spectrogram here. While it looks like continuous noise, like this big red blob of noise at nighttime, if you look at it over shorter time scales, you'll see that that blob is actually made up of lots and lots of, like, millions of little transient sounds. These are like pops, snaps, and clicks made by animals on the reef, similar to snapping shrimp noise. And there is some contribution from snapping shrimp, but there's also sounds from a lot of other animals. We know that a lot of invertebrates, fish, lobsters, sea urchins, hermit crabs, etc., produce these sounds. They've all been reported on individually, but we had this question: how do they contribute to the soundscape? And so there are some fundamental questions here. What is the effect of these creatures on the soundscape? And then looking at these things from a higher level, can we learn important ecological information from this ambient sound just using simple acoustic recorders? So going back to this spectrogram figure, this is a lot of information. How can we remove some of the information that doesn't matter as much? So abiological sounds, so sounds from boats and so on. You can't really see here, but sometimes they show up. The echoes, acoustic absorption from the seafloor, which depends on the frequency. So, if a fish made a particular sound and that sound interacted with the seafloor before it arrived at our hydrophone, the sound may not be representative of the sound we receive, may not be representative of what the fish made because it's been modified, sort of like speaking through a towel or something like that. So we want to try to account for those. And the tool at our disposal is an omnidirectional hydrophone that is a recorder, just like a handheld microphone, just records sound. You can't really tell where the sound came from, right? To do that is a lot more difficult. So the solution is what we call time gating and spectral analysis. And so I'll step through what that means over the next few slides. So it's mostly right. So if you look at the slide, you've got the spectrogram that I showed you on the left, and didn't quite come out right, but what I've done is I've taken a small sliver of that 24-hour spectrogram and I've expanded it out. So the horizontal axis now only covers 2 seconds rather than 24 hours. So in that short recording of sound, you see a very different picture. You see it's quite quiet now, and going by the same color scale as before, but that quiet is punctuated by these pulses that come through occasionally. And below that blue spectrogram, I have a time series, so raw output from the hydrophone. And you can see that for the most part, the signal is just, there's a lot of very low level static and occasionally these short, sharp sounds that arrive. There's a lot of energy contained in each of those sounds. So those are the sounds of interest. They're caused by biological processes, and they contain information that we want. So looking at one of those sounds, taking zooming in again on that time series, we see that sound identified by the sort of green pulse on the right side there. You've got the red and the blue, that's just background noise. Some of the red after the pulse may be echoes that come through afterwards, but we can try to just truncate the data and only look at that main pulse. If we take a spectrogram of that pulse, you can see on that left figure there, we're only looking at 10 milliseconds now. You can see that there's a blob in the area that's sort of between 5 and 10 kilohertz, that's on the left scale, that occurs, the left axis, that occurs between 3 and 6.5 milliseconds. It's still not very clear though, so we can zoom in even more. just looking at that green, that main pulse, that green area in that lower right plot, there we've taken a high resolution spectrogram, only 2 milliseconds. This red blob is much clearer. And if we take a line at around 5 kilohertz, cut through that spectrogram in the horizontal and plot that line on a figure by itself in the top right here, we follow that black line, you can see that lump, that peak that occurs around sort of 6.5 to 7 kilohertz. It's about 90 decibels, RE1, about 90 decibels in height. That's the mean height of that red bump. We can show that there's a peak frequency that's around 7 kilohertz. So basically, this is a long way of saying that one little click, pop, or snap has a center frequency around 7 kilohertz. And you can only find that out by zooming right in time, and you focus just on that moment that peak energy comes in, not when there are any echoes or not when there's background noise. So this is the way to time gate and look at the spectrum of those peaks of sound, and then we take that information, compile it with all of the other sounds that have come in, and we can say something about the distribution of sound producers in the immediate vicinity of the hydrophone. So this analysis technique was used to survey a comprehensive set of acoustic data that was collected around Hawaii. So this was done in conjunction with the Oceanwide Science Institute. There's a picture of Mark Lemmers there, who runs that organization. He's based in Maui. And this is work that I did, excuse me, under my graduate research program. And we collected acoustic data at all of these different locations around the Hawaiian Islands. You can see that there are some collections in the Northwest Hawaiian Islands that was courtesy of a NOAA rapid ecological monitoring cruise. But there was also other data collected around these other islands, the main Hawaiian Islands, that was all just from shore-based collection. So we surveyed many different environments in Hawaiian waters, short timescales, so only recording maybe 2 to 4 days of acoustic data. And this approach is similar to the survey approach that NOAA currently employs for their rapid ecological assessment of Hawaiian coral reefs. So these areas represented this incredible diversity of ecological conditions. There were some really degraded sites, like, for example, off of Kaloa in Kauai, just off a boat ramp. The visibility wasn't that great. There were some places like French Frigate Shoals in the Northwest Hawaiian Islands where these ulua were constantly in your face and they would get between your head and your hands so you couldn't see what you're doing. I mean, there's just incredible biomass and sort of health in that ecosystem there. So we collected information over this wide range of areas. In addition to the acoustic recordings, we took a lot of non-acoustic data, including this really interesting data set using remote camera intervalometers. So these are, if you look at this picture on the top right, there's a PVC frame that's been situated onto a piece of dead rock, and the frame carries 4 digital underwater cameras that are just facing down and looking at a patch of live coral. These images, these photographs, cameras would take photographs every few minutes until their batteries ran out, and they were programmed to turn on just before sunset. In addition, the standard NOAA rapid ecological assessment protocol were followed. Photo transects, you can see a diver there taking pictures of the reef, visual fish counts, and then measurements of rugosity, so how rough the sea floor is. And that measurement method involves just stringing a measurement tape over the sea floor, fitting it into every crevice and nook and cranny, and then taking another measurement tape and taking a straight sort of measurement from the start to the end and seeing what the difference in length is, right? The rougher the terrain, the longer the tape is that you've sort of stuffed into each nook and cranny. So just focusing in on those cameras for a moment, there's a zoomed image of one of the cameras, pretty inexpensive, but these things revealed a lot of really interesting information about the environment. Let me just exit out of this for a moment so I can play this video. I've got a video on the top right there of one of these time series, and you can see what happens. These photos are taken every 5 minutes. And you can see that at some point during this video, time-lapse video, the light level goes down. So it becomes nighttime, and you can see the strobe on the camera turns on. But what else happens is you can see that when night falls, all these little critters come out. You can see these hermit crabs and other animals crawling around on the reef. It's also interesting to note that there's more backscatter in the water, although that could just have been a function of the flash. So these animals aren't really there during the day, but when the sun goes down, they come out. And this was fascinating to us because the timing of the arrival of these organisms coincided really well with the increase in acoustic levels. So the animals inside each of these video frames were counted laboriously by a team of students and volunteers. And the data, such as the image shown on the bottom right here, was created. So on this graph, you have time of day, local sunsets, that vertical black line, and then the number of animals observed on the vertical axis there. And the types of animals are differentiated by color. So you can see that when the sun went down, everything came out to play slightly after. Lots and lots of shrimp, lots of crabs, and lots of hermit crabs in this particular location. And that coincides with that increase in acoustic energy. So these surveys yielded a huge amount of non-acoustic data as well as acoustic. So bottom cover, you can see the summary figure on the left. On the right, you can see individual organism count per frame per site of the same types of organism there. This is a really comprehensive data set, and it goes to support the acoustic data that was collected in the same locations at the same time. So we could make inferences between the two data sets now. So the number of variables here was enormous. And in addition to the amount of acoustic data, we're talking terabytes, right? So how can we make sense of all of this and show a sort of high-level image of what's going on here? And the answer to that is to perform what's called a principal component analysis. This back then, back in grad school, this was cutting edge to do this kind of work. It's fairly par for the course today, but a principal component analysis is a multivariate statistical tool to be able to look at relationships between many variables at the same time. It's something that I won't detail now, but I can show you how to interpret these things just at a very high level. So this is an output from a PCA. You have the first principal component on horizontal and the second on the vertical. Basically, if you see a relationship in space on this plot, there's a general association between the variables. So for example, here you see a cluster of some of these parameters together. You see sea urchins, rugosity, crusters, coralline algae, the prevalence of peacock grouper, etc., and reef cavities. They're associated with peaks in sound that are below 2 kilohertz. So I can say now that there's an association between a greater number of benthic organisms, their predators, and small-scale bottom roughness with low-frequency sounds. So it's a kind of high-level approach to interpreting this type of graph. At the same time, on the other side, there's some association between high-frequency peaks and fleshy macroalgae. They're sort of pointing in the same direction, right? And I think, yeah. So most of this association seems to be associated with sounds during the daytime as well. I'll talk about that in a moment. And lastly, I've circled the locations that were protected or semi-protected in green. Some were legitimate marine reserves, some were just protected because they were remote, and those are dashed ovals. And then conversely, the locations circled in brown are places that are impacted, jetties, boat ramps, harbors, etc. You can see that the the impacted sites kind of cluster to the left and the protected sites cluster to the right. And that suggests that there is a tangible difference in the soundscape between healthy areas and areas that are more impacted. So, the summary of that study is that reef soundscapes can tell us about various aspects of the ecosystem. Protected or remote coral reef areas sound quite different from degraded areas. The short sounds, the pops, clicks, and snaps produced by the multitude of animals on the coral reef that have peaks at lower frequency are associated with bottom roughness, greater numbers of benthic organisms, and their predators, like peacock, grouper, or roy. Short pops, clicks, and snaps that are of higher frequency are associated with the prevalence of seaweed, which was strange to us because seaweed doesn't make noise. So maybe there's some other relationship, and I'll talk about that a little bit more in a moment. So this is work that is summarized. This is a presentation that summarizes work that's published in a paper in the Marine Ecology Progress Series journal, and an image of the title and information about that paper is on the bottom right. So I want to talk a little more about this association between algae and sound. This intrigued us, and we contacted some of our colleagues at University of Hawaii Manoa. This is their Coconut Island campus. So on the left is Giacomo Giuli. He was a postdoc there, working under the late great Whitlow Owl. So Whitlow Owl is on the right. He passed away a few years ago, but he pioneered our understanding of sonar and dolphins. He worked for the Navy for a very long time. He was a great researcher. We used his lab to do some of our work. There's another guy who worked with us, Andy Haas. This is the only picture I could find of him on the bottom left, but he's now a senior researcher at a university in the Netherlands. But at the time, he was a postdoc at San Diego State University. He came and joined us, and he provided some valuable scientific instrumentation that was important for our work. So, at University of Hawaii, we wanted to delve a little more deeply into this association between sound and algae. And it just so happened that Coconut Island had this infestation of an invasive algae, Gracilaria salicornia, or they call it gorilla ogo. It's a particularly difficult algae to get rid of because it fragments. So you can just try and pull it out of the water, bits and pieces break off, and each piece becomes a new alga. So it's not particularly easy to get rid of. But there was a major invasive species mitigation effort underway where they were just vacuuming up tons of the stuff off the sea floor in Kaneohe Bay, and there was a bunch of this stuff available to work on. So we thought, hey, let's listen to it and see what kind of sound it makes, if anything. So here's a picture of me with some acoustic recording equipment doing an early experiment where we're testing this algae in these tanks, seeing if it makes any noise whatsoever. Things didn't really work out outdoors because it's just too loud out there. There's a lot of wind, other people walking around. We needed a place that was very quiet. So we ended up using a very quiet lab indoors. We also wanted to video the algae to see what it did. And this is, videoing it at a high resolution, looking for any small movements, anything that could explain if there were any sounds made, where those sounds came from. We made sure that there were no shrimp or anything else in the algae itself. So it was just the plants. So I've got a video here. I'll exit out again just to go to play this of what that camera captured. And this is just in an aquarium with seawater, and just equivalent to natural light. And the gorilla ogo is just sitting there photosynthesizing, doing its usual thing. So if you watch this video, you'll see, most of the time nothing's happening, but there are these little bubbles that are coming up from the algae. And it's almost like the water is carbonated, right? It's like fizz from champagne or soda or something like that. But these bubbles, they weren't just accidental. They weren't just bubbles that were attached to the seaweed when we stuck it in the tank. Those bubbles persisted for many hours. And we realized that the bubbles must be coming from the algae. Here we go. So, taking some high resolution macro images of the algae, it became clear that these bubbles were coming from the surface of the algae and sort of popping off the plant when they reached a certain size and floating to the surface. And this is something that happens not only inside tanks, but also in the wild. So on the left, there are some images from Floribeth Laval from the University of Hawaii. She took these in a surf zone of a turf algae covered in bubbles. And so this phenomenon happens in the wild as well. So we applied our signal analysis technique to the acoustic recordings from these tanks. So you can see on the left, top left, there's a time series where you just have static, that thick black line of just electronic noise. That's the noise limit of the recorder. And then occasionally you have these short, sharp sounds. Focusing on one of them, you can see on the top right there, it almost looks like ringing a bell. And you've got this nice sound, a nice sort of ping, and that sort of becomes quiet over time. And then you see an echo start just at the end of that image. Taking a high resolution spectrogram of a spectrogram of that time series results in the figure that you see as identified by number 3. And that's a figure that the vertical axis goes from 0 to 40 kilohertz. This is a different kind of recorder, and the horizontal is just 2 milliseconds, right? So it's a very short pulse of sound that's centered around like 13.5 kilohertz. And you can see in figure 4, a horizontal line has been taken through that spectrogram around sort of 1 millisecond, and you can see there's a peak frequency there. It's very clear, around sort of 13.5 kilohertz. So this is the same analysis technique that we use to evaluate coral reef sounds. We're using it to evaluate the sounds of algae. And it turns out that the mechanism associated with the production of the sound in those bubbles is quite clear. So, I mean, a simple experiment, we turn the light off and the bubbles stopped. We turn the light on, the bubbles started. And the longer the light was left on, the more frequent and larger the bubbles became. So on the left is a figure that shows elapsed time in hours on the horizontal, and then the peak frequency of bubbles in the vertical. And that gray bar represents when things were dark, so there's not much noise at all. But when the light was turned on, you can see that the heat map of bubbles becomes sort of more yellow than red, right? As time moved on, more and more bubbles appeared, and they were larger. So the peak frequency, well, then they were larger as well. I'll explain the relationship between frequency and size in a moment. On the right, there's a plot that shows sound exposure level. And this is the amount of acoustic, equivalent to acoustic work, right? How much work acoustic energy could perform. When the light levels were on, the amount of sound exposure level rose. When light turned off, it reduced. And that acoustic level followed fairly closely the concentration of dissolved oxygen in seawater. So there was this very close connection between oxygen and seawater. So, we were looking around for a causal relationship between bubbles and sound, and we found this wonderful publication that was a scientific paper that was issued in 1933 by someone named Minot, and it's titled On Musical Air Bubbles and the Sounds of Running Water. And Minot talks about how to characterize these bubbles just by listening to them. And he did 2 things in this paper. Firstly, he derived a physical relationship between bubble size and the frequency of its ping. So when a bubble is released from rest, it is momentarily perturbed, and it vibrates a little bit, like a bell. And when it vibrates, it creates a tone like a very short pulse. That tone has a specific frequency, just like a bell. And you could calculate what that frequency would be by looking at the size of the bubble through this equation here in the center of the slide. There are some parameters that you need to measure, like the density of the surrounding fluid, the pressure at the depth of the bubble, and also the polytropic gas coefficient. And so these parameters can be known. And so you can calculate pretty reliably the relationship between the radius of a bubble and its frequency. And so that's why I was talking about bubble size in that previous plot. As the frequency of a bubble decreases, it means that the size is actually increasing because frequency is inversely proportional to radius. Insightfully, Minot mentioned the humming of the sea in his 1933 paper. So that was a pretty clairvoyant thing for him to do. So anyway, what we did was we looked at these bubbles of oxygen coming off of these plants, which appear to be the result of excess oxygen produced by photosynthesis. We looked at the size profile of these bubbles over an hour and calculated what these sizes should be based on the acoustic recordings we made in the same tank. We overlaid those two distributions together, and you get that figure on the top left there, where the imaged bubble radii create that bar graph, and the acoustically derived bubble radii are depicted by that line, the black line. And you can see the distributions overlap pretty well. There's another plot on the left that shows that statistically they're the same. So we were able to show that photosynthesis produces sound in marine algae. So we know now that seaweed makes sound, and the mechanism is through the ringing of photosynthetic oxygen bubble separation, when those bubbles separate from the body of the algae. We can tell how much gas is being produced through Minot's equation just by listening to each bubble ping and estimating the radius of each bubble. And the sound correlates with algal cover over coral reefs. And we did this backwards. We first discovered that there was a correlation in the wild, and we thought, well, what is causing this? And we've found the solution here. And this is important because algae is a major indicator of coral reef ecosystem stress. And in the future, we hope that this type of discovery can be used to create systems that can quantitatively evaluate coral reef health just by listening to them. So this is this part of the talk has been an overview of a publication that came out in PLOS One. This is a 2018 paper that involves me, my wife, Giacomo, and Andreas Haas. So I will say that since then, we've gone on to do a lot more really interesting things to do with acoustics in Hawaii. That's the subject of another talk. I wanted to take a bit of time at the end of this this presentation just to talk about my present job and how it relates to underwater sound. So as was mentioned by Jill at the beginning of this seminar, I've moved on from working directly on underwater acoustics to a program director position at ARPA E, so the Advanced Research Projects Agency for Energy. This is part of the Department of Energy and was modeled after DARPA, the Defense Advanced Research Projects Agency, as an agency dedicated to creating transformative innovation through high-risk, high-reward research. So we recently launched a program called CCO2. That stands for Sensing Exports of Anthropogenic Carbon Through Ocean Observation. And this is a program that's going to begin in February 2024. Right now we're soliciting full applications. We have $45 million that we're managing to try to create viable measurement, reporting, and verification technology for marine carbon dioxide removal. So, there's this problem, climate change. We need to solve it through negative carbon industries, which is, not only do we decarbonize and we do things like bioelectric cars and use wind and solar power, etc., we have to create a negative carbon industry that is removing the CO2 we've already emitted in the atmosphere. And also, there are some industries that are really hard to decarbonize, like steel making. How can we continue those industries without contributing to global warming? So this negative carbon industry is what we're trying to create. The ocean has a lot of potential for absorbing a lot of the CO2 that's currently in the atmosphere. And there are means by which folks want to accelerate that process through seaweed sinking, through ocean ion fertilization. I mean, there are all these different techniques. There's a great report by the National Academies of Sciences on various carbon dioxide removal approaches. But we realized that none of these approaches will have any value in a future carbon credit market unless we can validate exactly how much CO2 they're removing and for how long. So that sensing capability doesn't exist yet. And it will allow, if it did exist, it will allow these ocean carbon dioxide removal technologies to have value in a carbon marketplace. So CCO2 is about creating physics-based, scalable carbon sensing technology in the ocean, and also some regional ocean models. This may seem somewhat tangential to acoustics, but I'm getting there. Please bear with me. There's going to be this emphasis on moving beyond today's methods of evaluating how much carbon is in the ocean and how carbon fluxes through the sea, through basically sort of chemical sensors, lab-based analysis of water samples that are collected, single point receivers. So these devices on the bottom left measure particulate organic carbon flux in the deep ocean, but only in one location, right? To go from that type of paradigm to sensors that rely upon, say, electromagnetic, acoustic, or optical methods to volumetrically sense how carbon moves through the ocean. So that's where the incorporation of sound comes in. This is just one of several ideas. There's an idea where we could potentially passively resolve ocean pH just using ambient noise. So it turns out that in the ocean, you have some boric acid naturally occurring in seawater, and there's an equilibrium reaction between boric acid and borate. That reaction is driven by pH, and it's also driven by acoustic energy. So when acoustic energy, well, it also impacts acoustic energy. So when sound waves propagate through the ocean, they impact molecules of boric acid, and the boric acid relaxes a little bit momentarily. And what that means is that these molecules change shape ever so slightly as acoustic energy propagates through the seawater. What that means is that causes some acoustic energy to be absorbed and released as heat, basically, in the ocean. And that means that the more boric acid there is, the more absorption there is in seawater. And with the boric acid equilibrium reaction is tied to pH. So as the ocean acidifies, there's less boric acid and the ocean becomes more echoey. So one could look at the attenuation of ambient noise, say near the surface, and compare that to ambient noise very deep in the ocean. And once you remove factors such as the natural attenuation due to geometric spreading, you can identify, you can conclude that the only reason for any additional attenuation is a change in pH. So, what could we learn from just listening to noise with regards to how carbon moves through the ocean? So this is one potential area of research that we would like to fund at ARPA E, and I'm looking forward to kicking off this program, CCO2, next year. So with that, that's my last slide. So thank you for listening, and I'll take questions.
Thank you so much, Simon. Wow, that is a lot of really comprehensive and detailed science. My coworkers and I were messaging each other on Slack, just like mind blown about everything that you're discussing. So to the audience, to those who are here and listening, please submit any questions into the Q&A box at the bottom of your Zoom screen. We do have some additional comments where you can listen to some sound clips that were collected as part of NOAA's Synced Sound project. So I'll just put those into the Q&A box so you're all able to access them. And here we go. I will read through these. Okay, to confirm, using octave level and/or third octave band analysis only can give you an idea of coral health by using algal cover as an indicator of health. No other manual analysis needed. We have PAM data from fall 2014 to present fall 2014 to present at various sites around Maui, including Kahekili. I believe
Okay.
that one's for you, if you can
Okay,
still, Simon.
yeah, I'm happy to comment on that. So coral or algal cover can only be inferred from the acoustic data. One would need to validate what's going on there. So, one way you could do that quite easily is by looking at changes over time. So if you are able to, if you have a recorder in a specific location, you get a spectrum of a certain type, and then you quantify the amount of algae. Later on, maybe a lot more algae shows up. If you then look at the spectrum again, and you can see that things have changed in the bands that I'm talking about, sort of in the higher frequencies, then you can infer something. So you sort of need to calibrate this approach at every particular site you may want to deploy it at. So if you have PAM data, that's potentially useful, but you would also need to find sort of non-acoustic environmental data to sort of compare it against. One of the things that I stress is that, from early on, we recognized the value of having complementary data sets that are time aligned, right? So you can see one thing in one set of data, see something in another, and then you can put them together. Yeah.
Thank you. And can you explain the carbon marketplace?
Sure. So I realized that I changed topics quite hard there, but I wanted to sort of talk a little bit about the current work I do. The carbon marketplace is something that's, well, actually, it exists in America already. So if you were, say, Microsoft, right, and you want to pledge that you are going to be zero impact to the environment, or you're zero emissions, you can't help but produce emissions as you, I don't know, employ people at a facility that uses power and has air conditioning and so on. So you offset those emissions by buying carbon credits. And you buy those carbon credits from groups or companies who do things like plant forests, right? And so planting forests is one example of carbon capture, and measurement, reporting, and validation is this act of going out and measuring how much carbon is locked up in those trees, right? So potentially you could measure how high the trees were, measure the trunk diameter, and infer, how much carbon has been trapped. So let's say you did that, and you could say, well, you could conclude that you sequestered a ton of carbon. Well, then you could sell that credit for 1 ton of carbon on the carbon marketplace, and a group like Microsoft would come in and buy it. But the critical issue there, though, is quality. So carbon credits have different qualities. If you just went and measured some trees and just guessed that you sequestered a ton of carbon, the quality of a credit would be really low, and it would be worth a very small amount of money. But if you had a comprehensive measurement and scientific protocol for evaluating how much carbon was sequestered, and you could say things about how long the carbon would stay out of the atmosphere, for example, then the credit would be worth a lot more. The quality would be high because the verification is more robust. And so, right now, if you can Google these carbon capture companies that are proposing to do things like sink millions of tons of seaweed in the ocean, or do ocean alkalinity enhancement by releasing a whole bunch of basic material into the ocean to try to sort of create this buffering capacity, right? All of these companies have business models that could work, but the quality of the credits that they're going to issue is entirely dependent on how accurate they can be in measuring their success. So that's the whole point of the CCO2 program.
Wonderful. I'm sure it sounds like you're staying very busy. So lots of work to be done.
It's a very fun job, I got to say.
Yeah, it's very creative and very technical. So we appreciate you, explaining all of it. I have a few more questions here as well off to the side. The principal component analysis, the one with all of the different relationships between, the time of day and sound and species, showed a lot of associations between sounds and species. Could we use soundscapes as a tool in our resource management toolkit? Could we hypothesize that if we were to create certain sounds in an area that we're trying to recover or recruit to, then other desired species might be attracted to it to settle or inhabit there?
Absolutely. So since we did this work, there's been some wonderful work by a guy named Steve Simpson from, I think, the University of Exeter in the UK. And there are some folks out of James Cook University in Australia that did some really, really good comprehensive work with larval recruitment and sound. And they found that, yes, if you play the sound of a healthy reef out of a loudspeaker that you put on a degraded reef, you will get increased recruitment of coral larvae, of invertebrate larvae, etc. Absolutely. They also found in the James Cook studies, they found that the soundscape was really important for survival post-recruitment. So there was some situation where if there was noise from some types of boats, but not others, there was increased mortality post-recruitment of little fish larvae, right? And it was almost like the fish couldn't hear the predators, right? So the soundscape really mattered for their survival. Yeah, so there's been a lot of work post this and looking at coral reef sound, but the dream of using passive acoustics to monitor the environment underwater has not yet been realized on a sort of regular basis. Like, we use satellite data regularly. We use diver collected information regularly. We don't use acoustic data just yet. And we're hoping that eventually that does happen.
That's a really good point. Where I mentioned earlier, we've been looking at potential sites for coral restoration. So it's good to consider, if it's noisier from boats or human use or interaction, maybe that site wouldn't be as beneficial or good for coral success as would be further away from some of those more popular sites, I guess. Another question from Meredith. I believe you mentioned that sounds can change during different moon phases. What would you expect to see during a full moon versus a new moon?
Yeah, so this is really interesting because we had seen literature that suggested that during a full moon, things are quite quiet because, animals tend to hide more when there's a lot more light, and there's a correlation between noise and light levels, well, inverse correlation, right? But it depends. The answer, like, we realize that it depends on where you are. So in some places during a full moon, things get really loud because there's in the summer, there's spawning, right? And so a lot of fish spawning is very acoustically active. In the new moon period, it's also very loud in some other places. So following this work, we deployed some recorders off of coral reefs in Bermuda. Bermuda is an interesting place because, like Hawaii, it gets deep really fast there. And, on one side, you've got this reef, on the other side, you've got the really, really deep ocean. And Bermuda is actually strikingly similar to Hawaii, except that it's in the Atlantic, right? We found that during the new moon, because it's so dark, there was a lot more noise from reefs that was, much more pronounced, much louder than during the day. So it depends on it depends on individual species and what the ecosystem looks like in that particular location. But there does certainly seem to be a relationship between moon phases and sound levels. We're hoping to publish that information soon, actually.
That's so exciting. Yes, okay, another note from Eden as well about collaborating for some coral restoration efforts. They are heading out, but we'll be in touch with them. So thank you, Eden, for coming in and providing some resources for us. I have a question out of my own curiosity. Some of those, there they're called spectrograms, right? The ones that you showed at the very beginning. How long does it typically take to process those? And are you doing it all just manually by hand, like you're looking at each little blip, or is there a program that can sort those out for you? Or how long does it typically take to process all of that information, all of that data?
If it's a single hydrophone, the data, if you've got a stream of data coming in, you could process it in real time. If you've got a log of data, like a memory card that has, say, a week's worth of acoustic recordings on it, it would probably take less than a week. Like, it's faster than real time. And I think it depends on how well one could write the software. I'm definitely someone who writes stuff that goes slowly, right? I'm not into optimizing things, but it's pretty straightforward. And when you asked a question about manual identification of peaks. So when we look at a recording of coral reef sound, there are many thousands of short, sharp sounds every minute, even every second in some cases, right? So automated detection and classification is really important. And because we're looking at short, sharp sounds, an intensity filter can do the job pretty well. Sure, it's not perfect, but it's just looking for the peaks and just sort of chopping out the signal on either side and just focusing on the peak is something that can be automated pretty easily.
Great. Yeah, that's very helpful. Okay, we'll do one more question just for fun, just because it's after 1 and I know that you are over on the East Coast. So thank you for hanging out with us. This one is a fun one from Amy, our executive director. I enjoyed it. So I can't help but think of the Jurassic Park scene with the T-Rex approaching the vehicle and the cup of water vibrating. Based on what you've seen, what creature would be the T-Rex of the underwater acoustic world? Does one big species ever show up and then everyone else goes silent, hiding from it?
So, I mean, I didn't talk about marine mammals, but they're pretty loud. In our work with coral reefs, we tend to view, especially in Hawaii, we tend to view the humpbacks as pests, right? Because you've got this nice recording all lined up, and then you've got this crazy whale song that comes in and just covers everything up, right?
Yeah.
That's pretty interesting. But I will say, when fish chorusing takes off, it's like an earthquake, right? It's so loud, and it's incredible because everybody is familiar with whale song, but people aren't familiar with the sounds made by these incredible choruses of fish that can be really loud and pervasive. So there are some very large sources of acoustic energy underwater, and they're mostly beyond the public eye, but all it takes is just putting a recorder down there, or even just going for a dive and like holding your breath for a moment and just listening, right? Especially at night, you can hear a lot of really interesting stuff.
That's incredible. We have so many speakers come on and talk about so many different aspects of the ocean and the different charismatic megafauna that we get to see out here, but really diving in and looking at the small stuff of the acoustics, I think, is something that we haven't really gotten to hear about before. So it just is very eye-opening and very, very cool to have us incorporate this now and put it in our management toolkit. So thank you for sharing with us. I'm going to go ahead and wrap us up here now that we're a little bit over time. So thank you for staying a little bit longer with us and for answering these questions. If any of you listening have any additional questions, you can send your questions to info@mauireefs.org and we can forward them on. If you logged on a little bit late, this presentation is still being recorded and it'll be available again on our Facebook and YouTube pages. But thank you so much again, Simon, for being here today.
My pleasure. Thanks for having me.
Okay. To all of our audience members, again, thank you so much for attending Maui Nui Marine Resource Council's monthly Know Your Ocean speaker series, sponsored by the County of Maui. Similar to Simon's talk today, our next speaker series event will be held on Wednesday, August 2nd at 5:30 p.m. via Zoom with Megan McElligott, and she'll be presenting on her study of underwater acoustics of spinner dolphins. So we just heard about how loud whales are. Spinner dolphins are also quite loud, I imagine. Her study focused on acoustics in Honolulu, Hulapo'e, and Manele Bays from 2021 to 2022. To sign up for this talk and any future talks, you can sign up for our free monthly e-newsletter, Reef and Brief, at mauireefs.org. You can also support ocean water quality on Maui by volunteering or donating to our community-based coastal water quality monitoring program, Hui Okawai'ola. This program is conducted by Maui Nui Marine Resource Council, the Nature Conservancy, and West Maui Ridge to Reefs Initiative in partnership with the State of Hawaii Department of Health Clean Water Branch. Our program leads and volunteers test ocean water quality every 3 weeks at 29 locations in South and West Maui. If you're a Maui resident looking for more volunteer opportunities, then this program is always seeking reliable West and South Maui volunteers. Visit huiokawai'ola.com for more information. And again, if you've missed some or all of today's presentation, you'll be able to view it on Facebook Live or on our Maui Nui Marine Resource Council YouTube page, Maui Reefs. Please follow us on social media as well for any updates and news. And last but not least, we want to thank all of our sponsors and supporters who are making our work possible. And we also wish to thank individual donors like you for donating your time to attend these webinars. Mahalo for making a difference. We will see you all next month. Mahalo for joining us tonight. We will see you soon. Have a great rest of your day. Aloha.